very interesting but also tiring day. I hope to give you some hope as a take-home message, because I think we are at an exciting moment now of developments in CF research which will really bring us therapy targeting the basic defects within a few years. So I'd like to share this with you during my next few slides. To start with this slide, which may be familiar to you, it's the slide of the survival median life expectancy in the United States based on the registry. And what you can see is here from the mid-80s on that there is a gradual increase of median life expectancy. So half of the people are expected to get older than, for instance, here, the age of 28 in the mid-80s. And it's gradually increasing up to now, during the last decades, about 37 years median survival in the US. But we see same kind of figures also in certain countries in Europe. I don't know exactly how it is in Greece, but I think eventually all countries will have the same pattern. Some maybe here, some maybe going there, but at the end there will be, and I'll show it in my last slide, not only this way, but also this way. I think our common mission is to improve the quality of life and prognosis for people with CF, all of us as we are sitting here. And we can do this by the patient organization, by stimulating international CF research, but from a patient's perspective, what are our needs that we have to address in our research questions and force research organizations, CF centers, researchers, pharmaceutical industry, biotech companies to go that way. You need an infrastructure for that. And of course it's not very well developed in every country, but with joint efforts we can see if we can come there. We have the patient's perspective, as I stated before. We have a research perspective and communicate between the two of them. From there, we can enforce a research network where also patients are involved, and I'll come back later to that. We can have funding programs, and tomorrow I'll also talk more about CF registries, which are very essential in this field. From that, we will get information for patients and peers, for instance, how now we are here, and also information for professionals and researchers to bring this further on, looking at new perspectives, and have this as a kind of cycle going on. So what's the process of drug development? I think it's good to realize that every drug that has been developed or will be developed has its own cycle and has a certain amount of time needed. It starts most of the time in the lab, in the laboratory, whereas drug discovery, preclinical testing, sometimes on animals, sometimes on cell lines. Then from there you get a moment where you go to what we call phase 1 trials. Phase 1 just answers the question, is it safe in humans? So often healthy volunteers are requested here to address this question. And to give an idea, from the about 500 possible leads that may lead to medication are here, maybe only 1 or 2 will eventually get here. So after this phase 1, when you can say, okay, this is safe, you can move on to phase 2 studies. And in phase 2 studies, it's most of the time a small number of patients where you address the question, is it likely to work in these patients? So smaller groups of patients are here involved. Sometimes different doses of medication is given, and just to see, is it working or is it not? If so, if you pass this phase, then you come to the phase 3 trials, and these are the large clinical studies where you can really say, okay, does it work or does it not? So often a lot of people are involved, where for instance you give 300 people the medication, 300 people don't know if they get the medication or not, and you can compare the 2 groups and you can say, okay, this is the effect. We see, for instance, an increase, substantial increase in FEV1, in lung function, or in BMI, nutritional status, or other parameters. After this, and I'll come back with some examples running right now, There's the phase of submission and approval. So in Europe, we have the European Medical Agency, also with involvement of patients, who will decide if this, based on all these studies and all the information they get from the pharmaceutical companies, can we approve it and can we say, okay, this is really medication that adds something, but which is also safe and where people can benefit from. Most of the time this process takes about half a year, and when EMA says, okay, this is safe and efficacious, then we approve it, and after that, in all countries in Europe, in principle, the medication could be prescribed. I say could be, because there is another phase here of reimbursement, not very important. So every country has this judgment of the EMA, and then goes in its own country starting to look, okay, can we reimburse it to our patients? And that is, I think, an issue which needs really a European approach, because it's very different in all the countries still. There are countries, for instance in Germany, when EMA says, okay, we approve it, for instance the next day patients would get it. And then during the next year, the pharmaceutical company has to show that it's really efficacious and it's really good for the patients. But, for instance, in the Netherlands, where I come from, well, this could take half a year, sometimes up to a year, before you really get it reimbursed. And there are countries who even take more time. But when it is approved and reimbursed, patients can get it. And then there's what we call the phase 4 trials, and that's when it's prescribed to the patients, they use it, but then you have to look at the continuity of is it really safe? Is it really safe on the longer term? And does it really work on a longer term? So that's what those kind of questions are addressed in phase 4 studies. So this whole time scale, it takes approximately 9 to 13 years in this until it reaches the patients, and the costs are enormously. Sometimes it's even exceeding, and especially with the programs who are developing now with very individualized therapy, these costs could be very high. Just a short reminder, because Christiane Knoops already mentioned it this morning, but I think it's good to really summarize for this talk. We have this CF gene defect where this protein CFTR is disturbed, which leads to an abnormal environment in the airways, which leads to obstruction of the airways, to infection, to inflammation, and to airway destruction. And here you get repeatedly vicious circles from here and from here to here, where you get a gradually decrease in your lung function and health status. Now, a lot of research has been focused until recently on this part, where you have the symptoms and try to relieve the symptoms. But fortunately, we see a shift now where more and more attention has been paid to this part. And this is very essential, because when we can intervene here in this process, you could prohibit all these negative sequences, and you can really target the basic defect in CF. And that's where we're aiming for. So I'll focus now in the rest of my talk on this part, where CFTR production is disturbed, and where medication has been developed, and also is being developed now, and is going to be developed in the next coming years to address this point. So there's a shift in the clinical trials direction, from treatment of symptoms to treatment of the basic problem in CF. And this is very important, it's mutation-specific treatment. So it's very important to know, for you or for your children, which specific mutation do you or does your child have. Because it can be very important or to participate in certain trials, or to get this medication prescribed. And we call it precision medicine. Sometimes also individualized medicine, personalized medicine, but now the term which is being brought up since this year is precision medicine. Just for you, in your individual case, which medication will suit you best. So we have this, the DNA in all our cells, and in part of each piece of DNA is a gene. And a gene is coding for blue eyes, black hair, but also for CFTR protein, yes or no. And what's a CFTR protein? It's just a string of all amino acids. They are put together, folded, and brought to the next level. And this folding is very essential in CF. This is a cross-section of the airway cells. So here's the airway. Here we have the cell layers. This is a healthy situation, and this is in CF. In the healthy situation, you have a very fluid mucus layer. So it's just a water layer where the cilia is moving all things towards the mouth. Because we have channels here, potassium channel, chloride channel, which can cross the cell membrane. Also water, because together this is salt, also water can just go across the cell membrane. However, in CF, there's a problem with this chloride channel. Chloride can go out. It's going to be together, so potassium will go in. And because there's salt in the cell, also water will go in the cell, which leads to less water here and very sticky mucus. That's very schematized the basic problem in CF. How does this protein now act in the cell? This is the chloride channel. We have this DNA. DNA is going to be duplicated in the cell, and it sends the message to build a good protein to the protein factory. In this protein factory, all the amino acids are put on the string, then are folded to be brought to the cell membrane, where it can attach to this chloride channel and make it to a good function. But in all these steps, something could go wrong, leading to a non-functioning chloride channel. We don't go into detail, but it's essential, and also mentioned this morning, that we have different classes of mutations in CF. So we have here again this DNA material, the protein fabric, and then going here to lead to a functioning chloride channel where salt and water is going in and out. Then we have these different classes of mutations. In class 1 mutation, there's a problem here in the reproduction of the DNA. And we call this nonsense mutations. And you can recognize them often when there is an X in the mutation. I know that in Greece, the proportion of patients with a class 1 mutation is higher than average than in other countries in Europe or in the US. So in Greece there are a lot of people also with this class 1 mutation. Class 2 mutations are the ones which is most frequent, F508del, where there is a replication of DNA, it's going to the fabric, but there's a problem with the folding. The protein can be folded and can be transported to the cell membrane, which leads to no chloride channels at all. In class 3 mutations, and I'll come back to that later, there's another problem. There are channels, but they have you can see them as a kind of door, but it's difficult to open them. So you need something to open that door. And this is, for instance, the G551D. I'll come back to that later. G551D, it's the first specific mutation where medication is available for now, called the Kalydeco. Just to finish this, we also have class 4 mutations. Then you have a functioning channel, but it's very difficult for the chloride to pass. But there is residual function, which is very essential for therapy, and I'll come back to that later. And this is, for instance, the I117H mutation. This is a typically Dutch mutation, A4503, but there are more of them. And finally, sometimes you have a distinction between class 5 and 6, but I keep it now into class 5 mutations. That's the one where there are very few channels, so the net result is that there's too small amount of water also here. But these two, 4 and 5, are also called the milder mutations, but bear in mind that this is in general. Each individual, and it was pointed out this morning, that there are multiple genes that are influencing the course of CS. So this is in general, you could say, these are the milder mutations, but in individuals it can be very different. There are people with worse mutations who do very well, and there are people with milder mutations who do less good. Why is this important? Because there are trials for these 7 classes of mutations. And I think especially for the Greek people with the class 1 mutation, normally it's about 10 to 15%, but I think in Greek it's up to 20% class 1 mutations. There was a very promising drug, ataluren, formerly called PTC124. And the phase 2 results, so in smaller groups, were rather promising. Then there was a phase 3 study, and the results were presented last year, in 2013, where overall, for the whole group who participated, children from 12 years and older, the whole group who participated showed not a really big effect. It was disappointing for everyone. Then they did some sub-analysis and saw that in the subgroup with patients who were never treated with tobramycin, when they got this ataluren, the effect was much higher. So they showed a significant effect on FEV1 of about 5 to 7%. So for the company, it was a point where they said, well, we're going to rethink for which people could this be helpful. And now a new phase 3 study is about to start for people who have never had tobramycin, children aged 6 years and older, and it's about to start, and the results are expected in 2015, beginning 2016. So we're really looking forward to those results. Concerning the class 2 mutations, which is about 19% of patients worldwide who have this class 2 mutation, so very important to find medication for this. Recently, the results for a phase 3 study from Vertex were published in about 500 patients, 12 years and older, where you saw the effect of Kalydeco, also called ivacaftor, formerly known during research as Vertex 70, which is a potentiator. And a potentiator does mean that it will especially open the door at the cell membrane. The combination of this Kalydeco with another Vertex lead, Vertex 809, also called lumacaftor, which is a corrector. It's folding the protein and bringing it up to the cell membrane. In studies, also in phase 2 studies and in the lab, it was shown that the combination of these two medications, in combination therapy, it resulted in 1 + 1 is 3. Separately, when you gave either this one or either this one, there was a small effect, but when you gave them both, the effect was tripled. So it's very important that this combination therapy, and we'll come back to that because it will be a key to other developments, this combination therapy that will be needed also with other kinds of medication to bring this forward. And the first results were presented this summer. It showed a significant effect on lung function of about 4 to 7%. Nutritional status improved, BMI and weight gain, and the time to the first exacerbation, which was pointed out this morning, was also much longer than in the people who got the placebo. Not mentioned, because the authorities like the FDA, which is the American regulatory authority, and EMA, which is the European regulatory authority, they focus a lot on hard parameters like lung function. But we know from people who participated in the study who are using Kaleido that they also experience a big improvement in their symptoms. For instance, people with their noses, sinuses were always very full. When they started the medication, it started drowning for 2 days, and afterwards it was clean. And we have anecdotal, it hasn't been studied in large groups, but we have information from patients who used it, who say, well, just after 2 days I got no coughing at all, no sputum at all. These are things that are very, very important, and also on the longer term, but haven't been really measured in these outcome parameters. So for future trials, we really have to address this. And for instance, when your lungs are destroyed, you can give Kaleido, but there will be not gain in lung function, but your symptoms could dramatically improve. And you really have stories of patients who were in hospital every 2 months, didn't go to school, and just after half a year of Kaleido were out, without symptoms, going to school, biking, going out, and even could leave their medication and therapy because they improved so good. So beneath these parameters, other parameters are very essential to look at. So the registration procedure for FDA and EMA will start this autumn, or has already been started. Vertex has prepared all documents. It will be sent in, and we expect from FDA and EMA, because they do it in a faster procedure, in the middle of next year, in the first half of next year, their agreement, hopefully, and then reimbursement procedures start. So we hope that it will be available at the second half of next year for patients, and those are patients with homozygous delta F508 mutations from 12 years and older, because the study has been performed in these patients, but already other studies are being performed now with younger age groups, and I'll come back to that later, also with other medications. But I think this is a very promising result, but a first step. But it will not be the cure to CF. And you see there's an improvement of about 4 to 7%, and there is improvement in nutritional status, but there is really a need to look at other leads or drugs who can have larger effects. And one of them is, for instance, Vertex 661, which is now in phase 2 study. So with the smaller groups, it has shown the proof of principle that this really works, and to a better extent than the combination therapy that has been performed. So phase 3 study is going to start on, to be designed on this part. It's really fresh, and there are also academic groups all over the world looking at new medication, new drugs, new leads which they could fit in. Biotech companies, smaller companies, sometimes related to academic groups, sometimes a part of it, are developing new therapies. And even big pharmaceutical companies like Bayer, Novartis, Pfizer, they are all in this process now of developing really targeted, mutation-specific therapies. And some of them are preclinical, but some of them are also entering now phase 1 in healthy volunteers. So concerning the class 3 mutations, which is about 4% of the people worldwide having these mutations, you may know that it was registered in 2013, the end of 2013, for patients with G551D mutation. For instance, in the Netherlands we have only one patient who's also using it now, but there are countries, and especially in it's a more Celtic mutation, so in the UK, in Ireland, in the US, there are much more people having this mutation. It is a potentiator, as we stated, it is available, and we are very happy that recently, earlier this year, the EMA also gave confirmation of registration for 9 other class 3 mutations, and currently they are in the national reimbursement process, and we hope to have this medication for these people available early 2015. In the Netherlands we have a Dutch project because we have 30 people with this mutation, which is a class 3 mutation, and which is also registered for Kaleido, but we just started a study with nutritional supplements because it is known from lab and from literature that nutritional supplements like curcumin in curry and genistein in soya beans also have a positive effect on the chloride channel function. So we are going to study this and also together with Kaleido see if this works well. And for this group, the combination therapy of Kaleido plus Vertex 661, as I just stated, phase 2 effect was demonstrated, and they're going on for further therapies. Just to show this figure, the effects of Kaleido on lung function. You see here people who participated from week 0 to 8, 16, 24, 32, 40 and 48. And here is the absolute change, the percent of lung function. And you see directly patients, these are the patients who got the Kaleido, and these the placebo group. And you see that directly when you start, you have an immediate effect of here, a gain of 10% in lung function, which is maintained over a long time. And there are already studies now of patients who are 3 years on Kaleido since the first studies, and they remain this gain. So there is, even after 3 years, there is still a difference. These people are gradually decreasing, and here you can see that these patients maintain their lung function, nutritional status, and the symptoms of less sputum that I told you before. And maybe in relation to the question this morning on Kaleido, when you have a lung transplant for your upper airways, it might be very helpful to have this Kaleido, because then a lot of people after transplant keep their ear and nose problems, but maybe you could address that point. So for the class 4 mutation, these are the patients with what we call residual function. And here we see in the lab that Kaleido and Lumacaftor also have a very positive effect. Also again, this combination, one of them separately has an effect, but when you give them in combination, 1 plus 1 is 4 or 5. But then for these people you really have to, for these patients you really have to see if it is a necessary treatment, because there are some patients with this mutation who are male that are infertile, but don't have any problems with normal lung function, normal nutritional status, normal life function. So you have to really look which patients could benefit from this medication. And this has further to be confirmed in studies, but they are under their way now. And I expect within 2 years also it will be a decision for these people. And the same accounts for class 5 mutations, where also residual function is about 5% of the patients. And here again, Kaleido and Lumacaftor have a very positive effect. And currently a study is going now on bronchodilators, because bronchodilators could have for some of these patients, especially these class 4 and 5 mutations, could also have the same effect as Kaleido and lumacaftor on the functioning of the CFTR protein. Just shortly to mention that in the Netherlands we have a research program called HIT-CF. It's a 5-year program of €4 million with 16 projects, and it aims at individualized treatment where preclinical and clinical researchers are working together with our foundation, and we want to predict the effects of combination of different medication in individual patients. And for that purpose we use a new test in the laboratory. It's called the organoids, and organoids are it's kind of mini gut system. So when you get a biopsy from a patient just with a thermometer in the rectum, just once you can get cells from the patient, you can bring them in the lab and have stem cells, and you can freeze them, hold them, freeze them, grow them as a mini gut, and you get these kind of figures. And the researchers in Utrecht, they succeeded to have a kind of flag on it to see if this mini gut system, the CFTR function, is working or not. So what you see in healthy people, this is a mini gut. There is fluid around the cell membrane, and this is in people with CF where there's no fluid and they are very shrinked. You can see it here. These are swollen organoids and these are shrinked organoids. And when you add the medication, for instance here in a patient with F5-Del and a class 3 mutation, when you do nothing you see that it's still very shrinked, but when you add Vertex 770 you see that they are swollen, so the CFTR function is working. What we can do now for each individual patient, you can take these cells and add all different kinds of medication. For instance, lumacaftor, but also the panel of the American CF Foundation, which has about 20 components, 20 possible new leads for medication. You can just add it in the lab to this individual patient and you will see, okay, this does work. When we add component 1, it doesn't work. When we add component 2, hey, it's working somewhat better. When we add component 3, it's working very good. So probably for each patient you will need an individual combination of therapy to have the maximum effect. That has the advantage that you don't have to try in every patient all these new medications, which is also of course expensive, which has a large burden for the patient just to try it, but that you can predict in the lab which combination of therapy would suit best for me or for my child. And this is another example where you see the same process. This is with a RLN7H mutation, which is somewhat better than the other one. And you see it's somewhat smaller, but when you add Vertex 770 Kaleido, you see a massive swelling. And they can measure the swelling with a computer and have an exact measurement. Oh, this is strange. Just short, how many times is that? Just shortly mention the drug pipeline, which is published on the website of the CFF. This is the one on the CFTR modulators. As we expressed, Kaleido is now to the patients. You see the different phases of research, and here when it reaches the patients. So Kaleido is now to the patients. Ataluren, which we discussed, was at the end of the phase 3 trial, but a new phase 3 trial will be performed. The combination therapy of Kaleido and this is lumacaftor has reached now phase 3. This is already finished and is going now in the registration procedure. And VX661 plus ivacaftor has ended now phase 2 and is going to phase 3. And this is another new one, but there are much more. This is from where the CFF is involved, but there are much more who are here in preclinical, but also phase 1 studies. I won't go into detail to all the other medications because time will be too short. This morning, Vincent already mentioned some medication like ibuprofen, other ones. This is a rather interesting one. Also in Belgium, a lot of research has been performed on it. It's also known as Viagra for other purposes, but it also has a really good effect on anti-inflammatory disease in CF. And also the anti-affective therapy. We have seen this morning, inhaled tobramycin, azithromycin, Caistin, Toby inhaled powder. We also have Colobrief now, Colistin inhaled powder. In principle, they are available for patients. This one was also mentioned this morning, almost available, Arikayce, Arovang, and here there are a few more. So really a lot of developments also on this one. Just shortly to mention gene therapy, because when in 1989 the CF gene was discovered, we all had hope that within 5 to 10 years we would have gene therapy. Well, we all know that it hasn't been succeeded since. But there was in the UK, the UK Trust spent lots of money to the consortium who went through on research for gene therapy, and about this time the results of a very critical phase 2 study are going to be presented. Maybe we hear somewhat more next week on the North American CF conference, if this in the form it is now could lead to gene therapy. We have to wait for that. And last year, on this stem cells, these organoids, it has been shown by a group from the proof of principle that they could repair. So in this stem cell minigut, they could take out the DNA, repair it, so get out the defect CFTR, put a kind of plaster on it and fix it, and they were able to grow new stem cells with the corrected DNA, but it will take, and this is the proof of principle, which is very good that it has been shown, but it will take at least 10 years before you can also say, okay, could we bring it back to people? How are the reactions then when you bring it back into the patients? Are there no side effects? So that really takes time, but the proof of principle is there. But many more years of research are necessary on the gene therapy. So in summary, we've seen several new candidates, especially targeting the basic defect in development. And I hope I made clear that combination therapy, different medication will be needed to target the basic problem in the cells, which are hopeful results, and which will probably lead to a prognosis, which we showed in the first slide was this, but with this new medication, these median life expectancy could rise much faster. I would like to finish with a statement that was made in last year's North American conference by Don Stevenson in a plenary lecture, and said it's no longer the question if CF will be cured, but when it will be cured. I think that's very hopeful to bring home. So let's move on with fostering the CF drug development in collaboration with patients, with doctors and with researchers to reach a common goal. We all want the elderly in CF. These are typically Dutch people, but the Greek variant will be somewhat different. And here I'd like to end and move to take any questions.
Thank you, Vincent. Thank you. I always admire Vincent for being able to explain very complicated stuff and bring it in a way that you feel you understand it. At least that's my feeling. I hope you have the same. Any questions about these new developments for Vincent? Next question. If you start working up here, I start walking to you, we meet each other in the middle.
Ευχαριστώ. Ήθελα να ρωτήσω, είδα τις κατηγορίες στα κλάσ 1 έως κλάσ 4 για τις μεταλλάξεις. Απλώς υπάρχουν κάποιες ακόμα που οι οποίες δεν υπάρχουν. Πώς θα μπορούσαμε να τις κατατάξουμε εμείς σαν γονείς? Δηλαδή δεν ήταν γραμμένες μέσα στα πλαίσια αυτά που έχετε ονομάσει. Αν είναι κάποιες από τις πιο άγνωστες, πώς θα μπορούσαμε να τις κατατάξουμε εμείς? Πώς θα μπορούσαμε να πληροφορείς για κάτι τέτοιο?
Yes, very good question and very important. There is a database of all the mutations, because the registries, I'll tell more about it tomorrow, all registries collect data and have sent it to America. There's a database now of about 60,000 people with all the mutations. And a research group is trying to sort out which mutations cause CF and which mutations are related to CF but doesn't cause many problems. So now, from the 2,000 mutations they are pretty sure that about 175 really cause problems in CF. The other ones, it is not clear. Or because there are too few to say something about it. There may be 3 or 4 mutations and they don't know how they do. And the other part is that, for instance, you can have mutations where people have normal lung function, normal nutritional status, no clinical problems. So that's one part. With that group, we are going to look now, and next month or next week will be the first meeting for that in the North American Conference, to look at all these mutations and also the classifications, because there are confusing and conflicting results in literature. Some say, okay, this is a class 2 mutation, where others say, no, this is class 4 or this is class 5. And from not all mutations, it is known yet. So the basic researchers in the lab are going to look, well, with this specific mutation, to what size would it fit? 1, 2, 3 or 4? Because it's important that for the new medication you could see, okay, it behaves like a class 1 mutation, so ataluren, for instance, would be a good medication for this patient, could be a good medication for this patient. But it's not sorted out completely. For some of them it's known, and there are articles, and especially also the group from Belgium, because the book has published on that it's clear, okay, this mutation belongs to this class. For some of them it's debatable, and for some of them it's not known. And for some of the mutations it's even not known if it really causes CF important problems or just smaller problems. Is that sufficient answer to your question?
Any other question?
There's one?
Sorry. Okay. Well, I start walking to you, you start walking to me, we meet in the middle.
I'd like to ask if this database is available on the internet for us to see.
Yes, it's the CFTR2 database. You can easily access it, and you can state at the beginning if you are a parent or a patient, or if you are a researcher or a clinician, because you can then get other information. Of course, you are allowed to go also to the researcher clinician information, but it's then more sorted out on a level that everyone could have a look on it. Maybe with CFTR2, I don't know. I'll see if I have it here. Sorry. It's but when you Google CFTR2 database, then you'll get there. It's a database and it's hosted by the Johns Hopkins Institute in Baltimore. And there it's also you also have to check some first rows that it is information that you cannot rely on the predictions that are made from it. It's just a general view, and it will not say that your child or yourself will just behave like this. It's all on a group level, but it gives you information. For instance, well, these mutations, patients with these mutations have a median lung function, because there are, for instance, 40 patients in it. They have a median lung function of 70% at the age of 18 years. Or a BMI of these patients is rather high. So you get really information on how the group of these patients with this specific mutation behaves. But notice, keep in notice that bear in mind that there's a large variation about it. It's just the means that they give, and there's variation about it.
We have one more question.
I would like to ask you, is there any limit of lung function, FEV1, that we cannot access to the new medicines? If you are 25%, maybe it's not worth it.
Yeah, that's a good question, and I think it's a point of debate. It hasn't been sorted out. You could discuss if you have a normal lung function, normal nutritional status, no symptoms at all, would it be necessary? That's one. On the other hand, you could have symptoms like I discussed the cases of people who had a massive sputum with normal lung function but had to do 3 times a day their exercises and nebulization, and they just could stop with it. That's an enormous impact. But there could also be patients where it would not be necessary. It has to be discussed. There's no clear sign on it now. But you could also think that, for instance, you could say you could have a start and stop criteria. You say, okay, we started, you have to meet certain conditions. We start and we're going to use it for a month because the effects are really dramatically fast. And see if you don't have any effects in 1 month, maybe you could stop them. And one other issue, if I may, is the newborns, because there are in more countries there are newborn screening programs. Would it be good to start in a phase where you don't have symptoms at all? Just to start there and avoid the negative cycle, lung function abnormalities, infections, inflammation, destroy of the airways. So that has to be sorted out.
I ask more about if you have very low lung function.
Very low.
20%.
Yes.
Maybe it doesn't want to pay the government, for example.
I don't think that will be the case, because we also have already examples of patients with very low lung function. They start with Kalydeco. For instance, they don't have an increase in lung function, but their symptoms are dramatically improved. And after that, for instance, BMI will increase. So that it's not just lung function. That's a good point.
Any more questions?
Καλησπέρα, είμαι μητέρα και ήθελα να ρωτήσω, μετά από τη καινούργια φάρμακα που θα έρθουν, που περιμένουν όλοι να έρθουν, η ψευδομονάδα θα συνεχίζει να υπάρχει σαν ψευδομονάδα, σαν ο διαβήτης συνεχίζει να υπάρχει, είναι κάτι που φεύγοντας η βλέμη μπορεί να αποκατασταθεί και το διαβητικό πρόβλημα των παιδιών μας?
Yeah, it's also a good question. There are already results from patients who have longer experience on Kalydeco, and sorry, you didn't have your. Yes, you were hearing. The patients who are already longer on Kalydeco, it was shown last year that the Pseudomonas disappeared from their sputum. They couldn't anymore from their sputum. So there's also a restoring effect on that. And of course, we need longer-term follow-up data, but it is really a successful effect on it.
Oh, one final question.
One final comment.
Sorry.
On the diabetes, there are no results yet on diabetes. There might be other problems with that, but it has to be sorted out.
Vincent, the example that you gave us regarding the patients where totally, the pseudomonas totally disappeared, were patients who have chronically infected of pseudomonas or not?
Yes, they were chronically infected. Yeah.
Okay. Well, we have come to a point that we have to end this session. Vincent, thank you very much for your ladies and gentlemen, now